Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the living organisms. And so far, what we have discussed about the classification of the living organisms, evolution of the living organisms, and then we have also understood the structure and function of the prokaryotic as well as the eukaryotic cell. And then we have also discussed about the structure and function of the different types of biomolecules. So we have discussed about the proteins, carbohydrates, and lipids, and as well as the nucleic acids. And in the previous module we have also discussed about the central dogma of molecular biology, or the central dogma of life, where we have discussed about the replication, transcription, and the translations. And in the current module we were discussing about some of the basic cellular processes. And in the uh, in the previous lecture we have discussed about the immunology, or the defense responses, and how the body or the cells are actually producing the different types of defense responses. And in the previous lecture we have also discussed about the cell cycle, cell growth, and as well as the apoptosis. Now in today's lecture we are going to discuss about how the cell is distributing the material within the cells, right? So you know that the cell is made up of the different types of organelles, right? We can have the mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies, and so on, right? Even nucleus also, right? So these organelles are getting the proteins and other biomolecules utilizing a very well developed vesicular trafficking system. So in today's lecture we are going to discuss about the vesicular trafficking system and how that is happening within the cell. So what is mean by the vesicular trafficking system? Okay, so you know that in the cell, for example, whether it is the animal cell or the plant cell, right? So if, for example, if this is a animal cell, so in the animal cell, what are the things we are going to have? We are going to have a nucleus, right, where we are going to have the so this is going to be a nucleus where you are going to have the DNA as a genetic material. Then outside this nucleus you are going to have the ER, right? So you are going to have the ER like this, right? So you're going to have the ER. Then you are going to have the Golgi bodies. So you're going to have the Golgi bodies, right? So uh, then you are going to have the mitochondria. So these are the different types of organelles what are present, right? And in a typical eukaryotic cell, in a plant cell also, you know, instead of mitochondria you are also going to have the chloroplast as well, right? Apart from that, you can also have the, you know, lysosomes. You can have the and this you know, and you can also have the double layer like plasma membrane, right? You can also have the plasma membrane. Now what is mean by the vesicular trafficking is that the proteins which are going to be produced by the ribosome. So you're going to have the ribosomes also, right? So the protein is going to be produced by the ribosomes. And you know that the ER is going to have the ribosomes docked on their surface, and that's why it is actually going to be called as the rough ER, right? And the protein is going to be produced through the ribosomes, and then these proteins are actually going to be distributed to the different organelles. For example, you can have the proteins which are present in the mitochondria. So mitochondria is actually going to have the protein from the two sources. For example, it can have its own synthesis, right? It can have its own synthesis, like utilizing the genome what is present within the mitochondria, and it can also have the import of the proteins from the cytosol, right? So these are the two sources. Now the proteins which are going to be import from the cytosol is actually going to go through a systematic delivery system, and that delivery system is nothing but a vesicular transport system. Now when you want to send the any packet, right? For example, if I want to send a packet to my home, or if I send a packet to my college, what I'm going to do is I am going to have a packet like, so I am going to have a packet like this, right? So this is going to be a letter which I want to send to IIT Guwahati, right? So how and the person who's going to send this packet is going to be a postman, right? So if you have a, or the postal services, right? But the how the postman will know that you are actually going to send the packet to the IIT Guwahati. So for that, what I am going to do is I am going to write the address here, right? This means if you want to do a vesicular trafficking, and if you want to do the vesicular transport, you require the two material. One, you require a person who can actually carry the packet, right? Which is going to be the postman in this case. And then you also require the address, right? So this address is going to be a unique address, right? Which means if I want to send a packet to the IIT Guwahati, even if I want to send the packet to my laboratory, it is going to have a unique address which is going to be pasted on this particular letter. And then only this packet is going to reach to my laboratory. How we are going to achieve in our daily life, we are actually going to be achieved that by the having a postman, and then going to have an address. And if you see a very systematic the address, like if you send a packet, right, you are going to add the address, right? And if you see the details of the address, okay, what you're going to see is I'm going to write my name, right? So that is going to be a unique, right? But there could be Vishal Trivedi in other places as well. So what I'll do is I'll then going to write the address, right? So it's going to be right like IIT Guwahati. Then I'm going to define which place. So I'm going to write Assam, right? And then I'm going to write India, right? Otherwise, so that is not still be unique, right? What I'm going to do is I'm going to write the postal code, right? If I write the postal code, right, for example 781039, then that has been a defined address, a very well defined address. So that is what going to happen. And then you also require a machinery, right? So you're going to have a machinery which is actually going to carry. So in this case, it's going to be the postman which is going to carry this packet, and the machinery is also going to have like the complete postal department where you're going to have the many people who are going to facilitate this process. Similar to that, when you want to send a packet, for example, a protein is being synthesized from the ribosome, right? So if the protein is being synthesized from the ribosome, and if it has to go to the mitochondria, right? Then what you are required is you are going to require an address, right? So you have to attach an address to this particular protein, which is going to be present in a vesicle, right? So this is vesicle is actually going to contain the protein of your interest, which you are going to send to the mitochondria. Now to this vesicle, we have to attach two things, right? We have to attach first the address, right? So in this case, if you see this case, we are actually writing each and everything in a alphanumeric codes, right? Whereas in the case of address, you are actually going to put some kind of tag here, right? You can put a tag onto this vesicle so that this tag is going to be recognized by the machinery, and that address is going to be unique. Similarly, you're going to have a unique tag which is going to be only for the mitochondria, and within the mitochondria also you can actually define whether you want to send the protein to the outer membrane of the mitochondria or to the inner membrane of the mitochondria, or whether you want to send the protein to the stroma or the cristae and all other places. So you can actually make the unique address, right? Apart from that, you also require the machinery, right? So in the machinery, here we have the machinery in terms of the postal services like the postman. In this case, you are going to have the machinery which has the multiple types of organelles which are going to facilitate this process. What are these organelles? You can actually require the help of the endoplasmic reticulum, you require the Golgi bodies, and you also require the lysosomes. Okay, so these are the three organelles which are actually going to participate into the vesicular transport, and that's why these organelles are also being considered as the organelles of the vesicular trafficking. So if you see, it's actually be very similar to what we actually practice in our daily life, and the same is going to happen in the cell as well, except that the machinery and as well as the way you are actually going to put the address is going to be different. Now so if you want to understand the vesicular trafficking, we have to understand the two aspects. One, we have to understand about the machinery and the organelle what are involved into this machinery, and then we also have to understand how this particular unique address is being generated and how that unique address is helping a vesicle to reach to the mitochondria. These organelles of the vesicular trafficking we have already discussed when we were discussing about the structure of the eukaryotic cells. So I'm not going to go into the detail of the each and every detail of this particular process, right? So the main function of the organelles of the organelle trafficking is to distribute the material throughout the cell. Three different organelles such as endoplasmic reticulum, Golgi apparatus and the lysosomes coordinately work together to maintain the vesicular transport of material across the cell. Eukaryotic cells take up the solid food from the outside through the process which is called as the endocytosis, whereas the uptake of the liquid is also called as the pinocytosis. This is also we have already discussed when we were discussing about the phagocytosis. Similarly, the material is secreted out of the cell, which is called as the exocytosis. In addition, the intravascular system delivers the protein synthesized in the endoplasmic reticulum to the different organelles. During endocytosis, the material present outside the cell bind to the cell surface through the cell surface receptor and trapped in a membranous structure which is called as the endosome. The endosomal vesicles are fused with the lysosome to form the late endosome, and in the late endosome, with the help of the lysosomal enzyme, material is digested and then the endosome is fused with the Golgi bodies and they deliver the content for the further distribution. In the similar manner, during secretion, the vesicle originate from the Golgi bodies and fuse with the plasma membrane to release the content outside the cell. So this is what is showing here, right? So this is the endocytosis where the food material which is present outside is going to be trapped into the membranous structure. Earlier it is going to form the early endosome, then it is going to form it is going to fuse with the lysosome, and that's how it is going to form the late endosomes, and then it is actually going to release the content. So the three organelles, so you can have the endoplasmic reticulum. So endoplasmic, you can have two different types of endoplasmic reticulum, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Rough endoplasmic reticulum is only the ER which is going to participate into the vesicular trafficking. Okay. So rough endoplasmic reticulum has a ribosome to attach to its and gives a rough appearance, where the smooth endoplasmic reticulum is devoid of the ribosome. So these are the rough endoplasmic reticulum where the ribosomes are attached onto the endoplasmic reticulum. The protein synthesis on the ribosome attached to the RER is sorted into the three categories such as the integral membrane proteins, proteins for the secretion, and the protein destined for the different organelles. So you can have the protein which are of three different types. It can be a protein of the integral membrane proteins, which means it will go to the plasma membrane. Then you can have the protein of the secretory pathways or the secretory proteins, and then you can also have the protein which is meant for the other organelles. Proteins are synthesized with a N-terminal signal peptide, and these signal peptides are recognized by the signal recognition particle on their target organelles. For example, if a protein is synthesized with a signal peptide for the mitochondria, it will attach to the signal recognition particle and receptor onto the outer membrane to deliver the proteins. The protein without any signal peptide is actually going to remain within the cytosol. These are the different functions of the endoplasmic reticulums. Then we have the Golgi bodies. So Golgi bodies are first visualized by a metallic stain invented by the Camillo Golgi, and it is made up of the flattened disc-like cisternae arranged in a stack manner to give three distinct zones. So it can have the cyst Golgi, medial Golgi and the trans Golgi. The cis Golgi receives the material or vesicle from the endoplasmic reticulum. So these are the cis Golgi, medial Golgi and the trans Golgi. Medial Golgi is the actual place where the proteins are covalently modified with the sugar. This is the anyway we are going to discuss when we are going to discuss about the the preparation of the label. And then we have the trans Golgi, and trans Golgi is the face of the Golgi towards the plasma membrane, and this site sorts the vesicle for their destinated organelle for the plasma membrane. So these are the different functions of the Golgi. It can actually help in the protein sorting, it can help in the protein modifications, and it can also help in the proteolysis. Then we have the lysosomes. So lysosomes are actually going to be discovered by the They are popularly known as societal bags due to their role in autophagy. A cellular process probably operates in the cell during starvation to meet their energy requirement. Lysosomal lumen is extremely acidic and it contains proteases, cytosolic enzyme, and it degrades the ingested material. So these are the functions of the lysosomes. Now, since we have discussed about the machinery, let's talk about the how we can be able to generate the label, right? So, trafficking signals. So, the glycosylation is the dire function of the biosynthetic secretory pathway in the endoplasmic reticulum and the Golgi apparatus. Approximately 50% of the protein characteristically expressed in a cell grow through this alteration, which involves the covalent addition of the sugar moiety to the specific amino acids. Mostly soluble and membrane-bound protein expressed in the endoplasmic reticulum undergo glycosylations, including all secreted proteins, surface receptors, and the ligand. Moreover, some proteins are being transferred from the Golgi to the cytosol are also glycosylated. So one of the easiest way of generating the trafficking signal is that you are actually going to modify the protein with the help of a glycosylation. Okay, and glycosylation means the attachment of sugar to the protein, right? And it's not only a single sugar, it's going to be a complex sugar molecule. You will know that we have the different types of sugar. We can have the glucose, we can have the fructose, we can have the mannose, we can have the even the galactose, right? So all these different types of sugar actually can come together either the individually or in a combination, and that's how it is actually going to make a complex structure and complex function, right? And these complex carbohydrate molecules are actually going to be recognized by the receptor which are present onto the target organelles, and that's how they are actually going to provide the address, right? So you can actually have a these sugar can combine with each other and they are actually going to give you a polysaccharide or a complex sugar, and that's how they are actually going to help you to generate the different types of organelles, right? Different types of trafficking signal sequences, and that's why they can be targeted to the specific organelles. As far as the glycosylation is concerned, the glycosylation can be of 4 different types. It can be N-linked glycosylation, it can be O-linked glycosylation, it can be C-linked glycosylation, and it can be a phosphoglycosylation. All these glycosylation types are being done by the whether they are what type of sugar peptide bond they are sharing and what kind of oligosaccharides are attached. So based on this, the glycosylation could be of 4 different types. So let's discuss about the N-linked glycosylation. So as the name suggests, the N-linked glycosylation is in this type of glycosylation, the glycans are covalently attached to the nitrogen on the asparagine or residues. This is the most common type of glycosylation. Approximately 90% of the glycoproteins are N-linked glycosylation. It occurs as soon as the protein is synthesized, rather almost simultaneously with the translational process. End-linked glycosylation process and the enzyme involved in this process are conserved across the wide variety of the species of the eukaryotes and the archaea. N-linked glycosylation can be broken down into several events. So how the N-linked glycosylation is going to happen? First event is the precursor glycan assembly. Then you're going to have the attachment. Then you're going to have the trimming, which means it's going to, do the final adjustments, and then there will be a maturation. So the initial step of glycosylations are identical for all protein. There is a difference in the trimming and maturation step, which generate the diversity in the glycosylated proteins. This means all these steps are actually going to be performed for the different types of protein, which are going to be the N-linked glycosylations, except that in the final stage, the trimming and the maturation step is going to be different, and because of that, it is actually going to generate the different types of trafficking signals. So the first step is that you can actually be able to produce the precursor glycan assembly, which means you are actually going to bring the different types of sugar and then you are going to put them together to generate a crude glycan assembly. The purpose is to assemble the 14 sugar molecule, which consists of the 3 glucose molecules, 2 N-acetyl glucosamine and 9 mannose sugar on the ER membrane via the dolicol. So dolicol is a phosphoisopenoid lipid carrier routed into the ER membrane by a pyrophosphate linkage. First, the 7 sugar molecules obtained from the sugar nucleotides like the UDP and the GDP sugars in the cytosol are added. After this assembly, the complex flipped to the ER lumen side and the 7 more sugars are added to form this complex sugar, and it is going to be attached to the dolicol, and that's how it is actually going to be considered as the precursor glycan, which means this is going to be a crude trafficking signal. Okay, so first you are going to have the crude trafficking signal, which is going to be generated, and where you are going to have the 3 glucose molecules, 2 N-acetyl glucosa molecule and 9 mannose sugar. And how it is actually going to be done? All these sugars molecules are going to be carried in the form of the UDP or the GDP sugar conjugates, which means the UDP and the GDP is actually going to provide the energy into the system, and that's why they are actually going to be work as a carrier. And after they are going to be assembled, they will be present into the ER lumen, and they are going to be form a crude trafficking signal. Once the crude is generated, then you are actually going to have the attachment of this crude sugar is onto the protein molecules. So the pre-assembled glycan is attached to the newly formed polypeptide chain via the enzyme which is called as the oligosaccharide transferase or OSTase. OSTase look for the consensus sequence like the asparagine X threonine serine, where X can be any amino acid except proline. Once it is recognizing, then the 14-mer precursor glycan is attached to through the carboxyamido nitrogen on the asparagine of the developing polypeptide chain. Beta hydroxyl alcohol group of serine and threonine residue act as a hydrogen bond donor for the reaction. So this is what is going to happen, right? So once the crude this sugar is going to be generated, right, they are actually going to be present onto the lumen side, and then it is actually going to generate the crude sugar molecules, and then these sugar molecules are going to be transferred onto the protein with the help of an enzyme which is called as the oligosaccharide transferase. And the dramatical arrangement of the glycan element, the glycan synthesis starts at the cytosolic phase of the ER, and when the structure is flipped into the ER lumen, then it is complete. Now the oligosaccharide transferase enzyme transfer the precursor glycan to the ASN on their nascent protein. So this is what is going to happen. Now this is actually a crude glycosylation. So after this is actually going to be trimmed so that you can actually remove some of the sugar molecules and you can actually also do the maturation. So then the next step is that you are going to have the trimming, right? So sugar molecules are trimmed off by the glycosidases via the hydrolysis. This process occurs both in the Golgi and the ER, and both serves as a different function. In the ER, the trimming is done both to screen the protein folding and specify when protein should be degraded. There is a continuous cycle of removal and addition of terminal glucose molecule that enable the protein folding. At first, the two terminal glucose are removed, followed by the binding of chylexine and chylitrenuclein to the chain via remaining glucose molecule. These then act as a chaperone for the folding of protein. Final glucose is hydrolyzed by the glucosidase 2 to the discharge of the glycoprotein from the chaperones. Folding output from this process has not yet acquired native conformation. Now, the UDP glucose glycoprotein glucosyl transferase act on the non-native folded protein and transfer a glucose to the glycoprotein, and the protein again is constrained to the lectin chaperone to add the protein folding. The cycle continues till the protein is properly folded. The properly folded proteins are trafficked to the Golgi bodies for the further processing, right? So those proteins which are not properly folded are identified by the ER resident mannosidases, and these proteins are deglycosylated by the glycan N and then forwarded to the ER associated degradation pathway. Apart from that, then there will be a glycan maturation, which means after that there will be trimming and all that. So this is what you're going to happen here, right? You're going to have a crude uh, the signal, and that crude trafficking signal is actually going to be matured by the different types of the enzymes, right? So you can have the mannosidase, you can have the mannosidase 2, and then you can also have the different types of processes, and through which this particular crude glycan assembly is actually going to be arranged and going to form a mature glycan assembly. So what are the things are going to happen? In the Golgi bodies, both trimming and the addition of sugar proteins take place, which generate diversity in the glycan structure of the glycoprotein. Up to previous step of trimming in the ER, the glycan structure of all protein is same. In Golgi bodies, each cisternae carry the specific enzyme and the stepwise process occurs, right? When the glycan is accessible to the Golgi bodies, mannosidase 1 and 2, it forms the complex oligosaccharides. These enzyme cuts off the several mannose residues and then get glycosylated by the glucose and N-acetyl glucosamine transferase, right? So in the first event, what you're going to do is the mannosidase 1 is actually going to remove so many mannose from this complex crude sugar, and then it is actually going to have the transfer of the glucose and acetyl glucosamine onto this by the help of the enzyme which is called as the glucose and acetyl glucosamine transferase, and the UDP is actually going to function as a carrier. So you can see that, right, the mannose is going to be removed, right, in some of the places, right, and N-acetyl glucosamine is going to be transferred. The same is going to happen when the mannosidase 2 is actually going to act, and that's how it is actually going to remove these mannose residues, and again some more molecules of the glucosamine, N-acetyl glycan, and all other sugars are going to be replaced. This process results in the formation of a common core region. After this, the several GTFs, multiple sugar moieties, are added to the core, which could be of variable length or could be of branched also. The complex oligosaccharide is resistant to the endoglycosidases like the glycan, and thus form the basis of the differentiating the two category of glycan. So this is the N-terminal glycosylations, where you are going to have the different types of steps, right? And that is actually going to so this is going to be called as N-linked glycosylation, which is mostly happening into the more than 90% proteins, where the first you are going to have a crude sugar moieties, right? We are going to have a crude sugar moieties, and then these crude sugar moieties are actually going to be trimmed and mature into the ER and the Golgi bodies, and that's how it is actually going to form a complex sugar complex, and that complex sugar molecule is going to be function as the trafficking signal, and that could be different for the different types of proteins, and that's how it is actually going to be targeted to the different organelles. Then we have the O-linked glycosylations. So the O-linked glycosylation is common for both the high molecular weight protein found in the mucus secretion and the proteoglycan core and then form the extracellular matrix. O-glycosylation is also common in the antibodies. Even though the N-linked glycosylation has already occurred in those proteins, it does not exclude the O-linked glycosylation. O-glycosylation is carried out by the enzyme which is called as the N-acetyl galactosamines or transferase, which transfer a single galactosamine N-acetyl galactosamine residue to the beta hydroxyl group of the serine or the threonine residues. Some proteins are glycosylated with the galactose N-acetyl galactosamine, some with the glucose, xylose and mannose and so on. It is based on which cell type and species glycosylation is occurring. The sugar moieties are obtained from the sugar nucleotides, just like in N-glycosylation, and the O-glycosylation forms a less complex structure compared to the N-glycosylation. So you can have the different types of sugar which are going to be come together, and that's how it is actually going to form a complex sugar which is going to be attached onto the protein molecules. Then we have the phosphoglycosylations. So this type of modification is limited to the parasites such as the Leishmania and Trypanosoma and slime molds like the Dictyostelium. It is abundant in the Leishmania and it is used to make the proteophosphoglycans, which is important for the organism to protect it from the host defense response. In this modification, the glycans are linked to the serine and threonine residue by the phosphodiester bond. Then we have the C-linked glycosylations, right? This is different from the other type of, it occurs in other, there is a bonding between the carbon nitrogen and the carbon oxygen, but the C glycosylation is characterized by the carbon-carbon bonding. This process is carried out by an enzyme which is called as the C-manosyl transferase, which links the C1 of the mannose to the C2 of the indole ring of the tryptophan. The enzyme identifies the precise sequence like the tryptophan, XX tryptophan, and they relocates amino residues from the dolichol P mannose to the first tryptophan in the sequence. C-manosylation has been spotted in the multiple cell lines and the rat liver microsomes. So with these glycosylations, it is actually going to achieve the vesicular trafficking. How it is going to happen? So in the vesicular trafficking, you can have the three different pathways. You can have the exocytosis, you can have the endocytosis, and then you can also have the distribution of the molecules within the cell types. Okay. And the three organelles which are going to participate into this, we are going to have the rough endoplasmic reticulum. It's going to be participate into the Golgi complex, and it's going to form the lysosomes. So in the endocytosis, the food material which are actually going to be trapped into the membrane bound structure, and these membrane bound structures are going to be called as the early endosomes, and these early endosomes are then going to be fused with the lysosomes, okay, and that's how they are actually going to form the late endosomes, and within the late endosomes, with the help of the enzymes from the lysosomes, it is actually going to degrade the food material into the constituents, monomers, and that's how it is going to be released. Apart from that, in the proteins which are going to be synthesized by the ribosome, which is attached onto the onto the ER, is actually going to be modified with the help of the different types of glycosylations, and these glycosylated protein are actually going to enter into the Golgi bodies through the cis Golgi. Once it enters into the Golgi, right, the Golgi is going to receive these vesicles, okay, and they are actually going to be further modified by the glycal trimming and as well as the maturation. And once they are going to be matured, then these vesicles are actually going to go out from the trans Golgi complex, and then from the trans Golgi complex, it is going to be packed into the vesicle with a signal for a particular organelle, right? So if the signal is for the mitochondria, it is actually go into the mitochondria. If signal is for the plasma membrane, the vesicle will fuse to the plasma membrane, and then it is actually going to release the content, and that is going to be exocytosis. So some of the, for example, some of the antibody which are going to be produced within the cell is actually going to be fused with the plasma membrane, and that's how they are actually going to release the antibodies into the circulation, and that's how they are actually going to participate into the immune responses. If the vesicles are going to have the no tag, right? So if the vesicles are not going to have any tag, then the vesicles are actually going to release the content into the cytosol. Same is true for the, if the vesicle has the signal for the mitochondria, they are actually going to release the content into the mitochondria. So this is all about the vesicular trafficking. What we have discussed about the machinery. So we have discussed about the endoplasmic reticulum, we have discussed about the Golgi complex, and we have discussed about the lysosomes. And then we also discuss about the how the vesicular trafficking is utilizing the different or how it is actually going to generate the addresses, like so that the address is actually going to be unique for a particular cell organelles. And in that process, we have discussed about the N-linked glycosylations. We have discussed about the different steps in which the N-linked glycosylation is going to happen, and how the ER and the Golgi complex are actually going to participate into these reactions, and how they are actually making a particular glycosylation pattern which is going to be unique for a particular cell organelles. Apart from that, we also discuss about the O-linked glycosylations, C-linked glycosylation, and the phosphoglycosylations. Now, if you are interested and if you are planning to read more about the vesicular trafficking, you can actually be able to generate, or you can be able to see that what are the different types of the vesicular trafficking signals are being generated by the ER and the Golgi complexes. So you can actually go to the web, and you can actually be able to see the glycosylation pattern for the for the mitochondrial signals, or the ER signals, or the chloroplast signal, and then you will understand that how the cell is actually generating these different types of complex sugar signals, and how specific it is. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more aspect related to the living organism. Thank you.